This section explains why retrieving a sequence from the NCBI nucleotide database is the entry point of the whole manual. Every later chapter begins with a text file, the reader produced here, so retrieval is the first step in the literal sense. The compression is this: a sequence has no home copy. INSDC keeps NCBI, DDBJ, and MBEL mirrored against each other, so identity is carried by the accession number, not by the file, and not by the server. The GI numbers were retired in September 2016 because a second identifier for one record is a liability. What the chapter actually teaches is the query grammar. A search reads, "Drosha" in the gene name field and "Bos Taurus" in the organism field. Search is a typed predicate over annotated fields, and the advanced builder is only a form that writes that string for you. Quoting a term collapses it to an exact phrase, and that usually costs you recall. This part explains what retrieval actually is when you ask a database for a protein. Retrieval means asking a question whose answer must be the same molecule you started from. The whole problem is that a protein has many names and one identity. UniProt KB gives that identity a stable accession, P00533, rather than the label EGFR, and every downstream tool in the book keys on the book, keys on that accession. So the operation is resolution. A fuzzy query, a gene name, an organism, a keyword, collapses onto one canonical record, and you export that record as FASTA, a header line plus the one-letter sequence, because FASTA is the only format the alignment and structure chapters accept as input. The chapter split between Swiss Prot and Tremble is the honest part. Swiss Prot is curated and human read, Tremble is auto-annotated and unread. Same retrieval, different trust. Everything after this chapter is a function whose argument is this: FASTA. This section explains why a chapter on downloading protein structure earns its place at all. Structure files exist because a protein's function is a shape, and shape data lives somewhere other than sequence data. The move here is to teach the fetch. Sequence databases hold letters. The Protein Data Bank, the PDB, holds coordinates. Each entry is one experimentally determined set of atomic positions, addressed by a 4-character identifier, retrieved as a flat text file whose lines are records such as atom, hetiam, and helix, rather than a FASTA string. The chapter's place in the book's arc is to hand you the raw material that the visualization and comparison chapters consume. Downloading is trivial. Knowing what you downloaded is what governs everything you may legitimately claim from it afterwards. One crystal, one resolution, one confirmation, possibly one of several chains. This section explains what a protein databank file actually holds, and why the many ways of drawing it are all the same data seen differently. A PDB file is a table of atom coordinates and nothing else. Every RASMOL rendering, wireframe, backbone, spacefill, ribbon, cartoon, molecular surface is one lossy projection of that same table, each discarding what the others need you to see. Backbone drops side chains to expose fold. Spacefill drops connectivity to expose fold. Spacefill drops connectivity to expose volume and packing. Cartoon drops atoms entirely to expose secondary structure. The measurement half of the chapter makes the identity explicit. The commands set picking distance, set picking angle, and the phi psi dihedral clicks read those same coordinates numerically. So image and number are 2 queries against one dataset. The display menu, therefore, carries the method. Picking a representation picks which question you can answer by looking. This section explains how a biological sequence stored in a file is never only its letters. A sequence in a file is letters plus a container. Grammar, where the identifier lives, whether residues are numbered, what marks a gap, what terminates the record. Chapter 5's thesis is that these grammars are arbitrary and program-specific. The practical unit of bioinformatics work is therefore the sequence in a format, not the sequence in a format, not the sequence, and every downstream tool imposes its own. Hence, we seek one function, format in, format out, the residues invariant under it. FASTA marks its header with a greater than sign. GenBank uses locus and origin. Pilot puts a species count on the first line. GCG divides with a double dot. Different envelopes, same content. The IUPAC codes are the fixed point beneath all of them. The alphabet no conversion may touch. This section takes the suite program by program and names the string operation behind each one. Every program in the suite is a string operation wearing a biological constraint. Filter DNA is character class deletion. Reverse complement is reversal, composed with an involution on the IUPAC alphabet. Range extractor is slicing. DNA stats is a histogram over 4 symbols, and codon usage is that same histogram over the 64-symbol alphabet induced by reading frame. Pattern find is regular expression matching, and its default pattern, CTT, followed by either C or A, admits it outright. The chapter's function in the book's arc is to hand you the operator basis before the algorithms that compose it. Alignment, BLAST, and restriction mapping all reduce to these moves under a scoring rule. Biology enters only 3 times: which alphabet, which codon table, which substrings an enzyme recognizes. This section explains what a restriction map is and why finding enzyme sites is the operation that makes a sequence physically manipulable. A restriction map is a coordinate system that the enzyme catalog imposes on your sequence. Every recognition site is an address where a cut is possible, so finding the sites is finding where DNA can be surgically opened. This chapter teaches one instrument, NEB Sutter, and treats it as the general case. Paste the sequence, declare the topology as linear or circular, choose an enzyme set, and read back a map, and linear or circular, choose an enzyme set, and read back a map, annotated with cut positions. Overhang DNA can be surgically opened. Its place in the book's arc is instrumental. This is the last chapter of the single sequence section before section 2 turns to alignment. A sequence has been fetched, formatted, and translated. Now it is cut, and the cut is what makes it manipulable in a wet lab. Cloning, RFLP, restriction mapping, and the RE assay are each listed as uses of the chapter, and each one asks the same question for a different purpose. Does this enzyme cut here or not? This part explains why the dot plot comes before alignment proper. A dot plot puts one sequence on each axis and marks every cell where the residues agree, with no scoring, no gaps, and no dynamic programming. The whole comparison matrix is shown rather than one optimal path through it. Structure then reads off geometry. An unbroken main diagonal means identity. A diagonal shifted sideways means an indel. Parallel short diagonals mean repeats. An anti-diagonal means inversion. Window size and mismatch limit are the only knobs, and they trade signal against noise. With 4 bases, the noise floor is high, so small windows drown the picture. The chapter's place in the book's arc is preparatory. Look at the alignment landscape before committing to an algorithm that returns a single answer, which is where chapters 9 and 10 go. This section explains why global alignment is a shortest path problem. Every possible end-to-end alignment of 2 sequences is a monotone staircase path through a lattice of n+1 rows by n+1 columns. Diagonal steps core depends only on its 3 predecessors. Horizontal and vertical steps consume 1 character and pay a gap. That reframing is the whole chapter. An exponential search over gap placements becomes the shortest path. An exponential search over gap placements becomes the shortest paths, and shortest paths obey man toppers dies. To each cell's best score depends only on its three predecessors, horizontal and vertical steps. Horizontal and vertical steps consume one character. The recurrence is this. F of I and j equals the maximum of three quantities, the diagonal cell plus the substitution score S of I and j, the cell above plus the gap penalty d, and the cell to the left plus the gap penalty d. Score forward, trace back, and the arrows are the alignment itself. Ties fork into multiple optimal paths, which is why backtracking follows every contributing cell. The scoring scheme is an input, not a truth. Change +2, -1, -2, and the optimal path moves. This section explains why Smith-Waterman differs from global alignment by exactly one operation, zeroing a cell. That single truncation is the whole algorithm. Needleman Wunsch forces every residue of both sequences into one alignment, so a long stretch of noise flanking a conserved motif drags the optimal path through it. Clamp every negative score to 0 and each cell stops carrying debt forward. A bad prefix costs nothing, so any cell can serve as a fresh origin. The best local alignment is then the matrix maximum, traced back until the score hits 0. Same recurrence, same scoring scheme, one truncation. The matrix stops encoding how the whole sequences correspond and starts encoding where correspondence pays. Chapter 10's own contrast, in Table 10.1, puts the difference entirely in the traceback. The local traceback begins at the matrix maximum and terminates at the first 0, where the global traceback begins at the corner cell and terminates at the origin. This section explains what changes when sequence alignment arrives as a service rather than as something you implement. The alignment algorithms are already established, and the dynamic programming machinery behind them is now reached through web forms. What you gain is databases. What you supply is a query. The move that carries the chapter is that the tool choice encodes the biological question, pairwise BLAST, when you already know both partners. Database BLAST when the second sequence is the unknown. Multiple alignment through Clustal when the object of interest is the conserved column rather than any single pair. The scoring substrate stays the same throughout. Substitution matrix, gap penalty, e-value. All of it is surfaced as parameters that ask to be read before they are accepted. This section explains what Blaston gives up and what it buys in exchange. Chapter 10 handed you the optimum. Smith-Waterman finds the best local alignment and pays quadratic time for it, which makes it unrunnable against a database of billions of bases. BLAST trades the guarantee for a seed. The seed is an exact word match of fixed length extended outward in both directions until the score decays. Everything on the NCBI form is that one dial exposed. Word size sets how much similarity you assume before looking, with Mega Blast assuming 95% and Blast assuming 95% and Blastin assuming little and paying for it in time. The match to mismatch ratio and the gap cost shape the extension, and the e-value judges what survives. The chapter's real content is learning to read a search whose answer is statistical. This section explains how the 4 BLAST protein programs relate to each other. The 4 programs are one algorithm wearing 4 different scoring matrices. Plain BLAST P scores every alignment against a fixed substitution table, PAM for close relatives, BLOSSOM for divergent ones, so its reach is fixed before the search begins. The other 3 build the matrix from the search itself. PSI, BLAST harvests its own hits into a position-specific profile and requeries with it. PHI BLAST anchors that profile to a declared motif. Delta, BLAST seeds it from the conserved domain database before the first pass. Sensitivity is a function of where the scoring model comes from, and the chapter's real content is that ladder. Everything else is where the checkbox sits. This section explains why BLAST-X belongs to gene prediction rather than to alignment. Nucleotide identity decays under silent substitution. Amino acid identity survives it. BLAST-DX buys that conservation with compute. The nucleotide query is translated in all 6 reading frames, and each translation is searched against protein databases. 6-fold cost, 1 structural dividend. The frame that returns a hit is the open reading frame. A similarity search thereby answers a question nobody posed to it. Does this sequence code for protein at all? And where does it begin? That is why the chapter files BLAST-X under gene prediction rather than alignment. Interpretation collapses to a single line. Everything reads exactly as BLAST-EP does, except the frame field, and frame carries the entire discovery. This section explains the single inversion that defines T-BLAST-N. You bring a protein query to a nucleotide database, and the translation happens on the database side. BLAST-X translates what you bring, T-BLAST-N translates what you search. 6 frames are generated per subject sequence, so an unannotated genome or an EST pile becomes searchable as protein without anyone having called a genome, or an EST pile becomes searchable as protein without anyone having called a gene in it first. That accounts for the target databases, the chapter names, EST and HTGS, precisely the records nobody has annotated. The pairwise output carries a field called frame, which of the 6 reading frames the hit landed in, and that single field is what distinguishes reading a T-BLAST-N alignment from reading a BLAST-TV1. Annotation is deferred to the alignment rather than presumed by the database. This section explains what T-BLAST-X does and why its design follows from the state of the data it is aimed at. 6 frames on each side, a protein alphabet, and nothing trusted as annotation. That is the whole of it. T-BLAST-X translates the query, and every database entry into all 6 reading frames, then compares amino acids to amino acids. The reason is that a sequencing error, a wrong base, a frame shift, destroys a nucleotide match locally while leaving protein similarity detectable in some frame. Coding sequence is conserved at the residue level long after the codons diverge. So the tool is aimed exactly where annotation is absent, and errors are dense, single pass, ESTs, draft genomes. The cost is combinatorial. 36 frame pair comparisons per sequence pair, which is why the chapter's only warning is against large queries. The extra output column, N, counting ungapped segment pairs, is that geometry surfacing in the result. This part explains what a primer actually is at the level of the reaction. A primer is a thermodynamic boundary condition you impose on the reaction, and the sequence is what that condition happens to spell. Every rule in the chapter is one constraint expressed in different coordinates: 18 to 25 nucleotides, 45 to 55% GC content, a melting temperature within 2 Deg-C ceilings. This deltius of its partner, 2 G ceilings, among the last 4, the single requirement underneath all of them is that the duplex you want must be the most stable pairing available at the annealing temperature, and every competing structure, hairpin, self-dimer, cross-dimer, must be less stable than it. Table 17.2 states this directly, as delta G ceilings. Specificity and efficiency are the 2 failure directions of that one margin. Too little stability and nothing anneals. Too much and the wrong things do. This section explains how primer 3 actually works, and why its output is a starting point rather than a finished answer. Primer 3 is a constrained search, not a designer. It walks every substring of your input sequence and ranks them by a weighted penalty function whose weights you set yourself. Chapter 17 supplied the constraints, length, melting temperature, GC percentage, and self-complementarity. This chapter's whole content is that specifying those constraints is itself the decifying those content is. That specifying those constraints is itself the design act? The trap. The chapter states plainly is what happens when you leave a parameter blank. The software silently substitutes its own default. An unset constraint is still a choice you made without knowing you made it. The output is 5 candidate pairs, not an answer. The tool optimizes exactly what it was told and stays blind to everything else. That is why the pairs go on to IDT, a LIGO analyzer, which scores the hairpins and dimers Primer 3 never looked at. This section explains why primer quality checking is a rejection stage. A primer earns its place by surviving falsification. Chapter 19 is where candidates are rejected and every tool in it computes one number, delta G, the free energy of a structure you do not want. A hairpin, a self-dimer, a heterodimer, and an amplicon fold are all the same physical event at different scales, the oligo binding something other than its target. One thermodynamic threshold therefore ranks all of them. More negative means more stable, and more stable means worse. Specificity gets the same treatment through primer B last. A match anywhere along the sequence is tolerable. A match at the 3' end is fatal, because that is where polymerase extends. The whole content of the chapter is the sign convention, plus a table of cutoffs, SYBR. Green reports the presence of any double-stranded DNA. This section explains why that single property forces every discrimination decision upstream into the primer pair. A dimer, a hairpin, and a spurious amplicon all illuminate exactly as the intended product does. The dye contributes 0 sequence discrimination. The entire specificity budget of the assay must therefore be spent upstream inside the primer pair. Table 20.1 itemizes that debt across 15 parameters. Length and GC content bound, mispriming, runs of identical bases and 3' delta G suppress self-structure. Primer. TEP-A. PPLA. The tell that this is a budget and not a checklist sits in row 2. Probe chemistries. Buy discrimination a second time at the probe and so tolerate a shorter amplicon 60 to 150 base pairs against 120 to 200 here. This section explains what the UPGMA tree construction actually produces. UPGMA imposes ultrametricity on the data. The procedure itself is plain greedy, agglomerative clustering. Merge the closest pair, replace it by a group whose distance to everyone else is the arithmetic mean of its members' distances, then repeat. Each merge is drawn at half the merging distance, so every leaf finishes equidistant from the root. Equal depth encodes the molecular clock you assumed going in. It corroborates nothing. The chapter's anchor is the non-ultrametric sidebar. Hand the same arithmetic, a matrix that violates the 3-point condition, where the distance from A to C is greater than the maximum of the distance from A to B and the distance from B to C, and it returns a fully drawn tree of the wrong topology. Nothing in the output distinguishes that run from the good one. This section explains how the Fitch-Margoliouth algorithm builds a phylogenetic tree and why it differs from the greedy merging approach. The method is least squares against the whole distance matrix. where UPGMA collapses the closest pair and averages away the evidence, Fitch-Margoliouth keeps every pairwise distance as a constraint and picks the tree whose additive branch lengths minimize the residual, observed minus fitted. The trick that makes it tractable is the composite taxon. Freeze one pair against everything else lumped together as a single group called X. Solve the resulting 3-point star exactly. 3 equations, 3 unknowns, giving branch lengths P1, Q1, and R1. Then expand X and iterate. No molecular clock is assumed, so branches may run at different rates. Additivity alone carries the reconstruction. The cost shows at the extremes. Homoplasy is invisible, so long distances come out short, and rooting needs an outgroup bolted on from outside. This section explains the neighbor joining method for building a phylogenetic tree from a distance matrix. 2 taxa can look adjacent in that matrix merely because both evolve slowly. Neighbor joining strips that confound before it commits. Each pairwise distance is adjusted by how far both partners diverge from every other node, and the pair minimizing the corrected value is adjusted. Start from a star, all taxa on one node, and decompose it one join at a time. Each join collapses a pair into a composite, shrinking the matrix by 1. The procedure is greedy, and a join is never revisited. What the correction buys is minimum evolution, the shortest total branch length, with no clock assumed and branches free to differ in rate. The chapter's primitive reaches well beyond phylogeny. A pairwise score carries no information until it is normalized against the ambient set. This section explains what the maximum parsimony method does differently from every tree-building method before it. Parsimony inverts what a tree is for. The distance methods preceding it, UPGMA, neighbor joining, and minimum evolution, crush each sequence pair into one number and cluster on that number. Parsimony refuses that collapse. It keeps every alignment column as its own character and scores a topology by how many substitutions that topology forces. So the tree is ranked against the data rather than inferred from it. Enumerate topologies, cost each one, keep the cheapest. Mukhopadhyay's word for this posture is cladistic, as against phonetic. The evidence is shared derived state, and summed dissimilarity has no role in it. The cost of that posture is combinatorial. 4 taxa give 3 unrooted trees. 50 taxa give 2.84 times 10 to the 74th power. That growth is why the chapter ends where it does. Branch and bound arrives as a structural necessity of the search space itself, carrying the weight that an optimization refinement would elsewhere. Branch lengths vanish from the output. Nothing metric was ever computed. This section explains how the minimum evolution method builds a phylogenetic tree. A tree becomes the argument that minimizes a single number. Sum every branch length, then keep the topology where that sum is smallest. Distances are the only currency here. The alignment collapses into a matrix of pairwise mismatch counts, and which site differed is discarded before the search even starts. That discard is the whole trade. Minimum evolution buys speed and an unrooted tree from an optimality criterion computed on numbers alone. Maximum parsimony pays per site bookkeeping to keep the positions, which is why its branches carry site labels, while minimum evolutions carry only counts. The 4-sequence example, sequences S1 through S4 with distances running from 3 to 7, demonstrates the criterion mechanically, cluster the closest pair, check the 4-point condition. It is a demonstration of the criterion, not any real topology search. This section explains what a phylogenetic tree is actually built from, and why every stage of the MEGA-7 procedure exists. The tree is computed from a distance matrix. Sequences enter the chapter only as raw material, and the whole procedure is the chain that manufactures that matrix honestly. Alignment comes first, because a column is the only unit in which two residues are claims about the same ancestral site. Unaligned sequences have no comparable positions, so clustal W or muscle is the step where homology gets asserted, and everything downstream inherits that assertion. Then a substitution model turns column differences into expected substitutions, correcting for the changes that overwrote themselves. Then neighbor joining reads the corrected distances back into topology. Bootstrap resampling of columns measures one thing only: how reproducibly the columns support a branch. Chapter 26 states it flatly that this bears no relationship to the accuracy of the tree. This section explains what a phylogenetic tree actually measures. A tree drawn from sequences carries less information than it displays. Only the horizontal axis is measurement, branch length in substitutions per site, and even that saturates. A branch length of 1.0 means 100 substitutions spread across 100 sites, not 100 change sites. The vertical axis is placement alone. Any subtree rotates freely about its internal node, and the tree means the same thing as figure 27.2 shows. Rectangular, straight, radial, and circular are one object under 4 projections. The names declare only what branch length is denominated in. A cladogram denominates in nothing, a phylogram in genetic change, a phenogram in resemblance, a chronogram in time. So reading a tree reduces to sorting its visible degrees of freedom into 2 classes: gauge and measurement. This section explains how a protein's local fold is predicted from its amino acid sequence alone, and why one kind of structure is far easier to predict than the other. The chapter is a lab protocol rather than a theory. Chapter: 1 mechanism, 2 tools. The whole claim is that guessing local folding from sequence works because helices and sheets each impose a statistical signature on the residues that can occupy them. Helices for each residue, ene, glutamate, leucine, and methionine, and are broken by proline. Loops accumulate charged and polar residues at the aqueous interface. The procedure is: read a window of sequence, score it against those propensities, and emit H, E, or C for each residue. Here is the asymmetry that matters. Alpha helix prediction is easy because its constraint is local. Hydrogen bonds form between near neighbors, and there is a hydrophobic period of roughly 3 to 4 residues along the chain. Beta sheets pair strands that may sit far apart in the sequence, so the evidence for them is not contained in any single window. That is why PSIPRED reports a per-residue confidence bar rather than a verdict, and it is why the chapter closes by asking what constrains beta sheet prediction. The answer is non-locality. This section explains how a protein's 3-dimensional shape gets predicted when its sequence is all you have. Fold conservation outruns sequence conservation, and that gap is the whole method. 2 proteins descended from a common ancestor drift in their residues far faster than they drift in shape. So a solved structure taken from a relative is a better estimate of your unsolved protein than any physics you could run from scratch. The chapter's anchor is deliberately easy. Buffalo pancreatic ribonuclease, never crystallized, modeled on the bovine 1 PDB4RTE at 95.97% identity. Swiss. Model is only the arm that performs the transfer. The complementary case, no homolog available, falls back to ab initio energy minimization over conformation space. The whole practical of homology modeling exists because that fallback is expensive and unreliable. This part explains why threading works at all, and the answer is an asymmetry. Fold space is finite, where sequence space is not. When no homologue exists, similarity search has nothing to match against. So threading abandons sequence comparison entirely and slides the query directly onto each candidate backbone in a library of known folds, scoring the fit by an energy function rather than by residue identity. Recognition and prediction collapse into one step. The template that accepts the sequence at lowest energy is itself the answer. The chapter sits between homology modeling and ab initio methods, the middle rung, structural evidence still borrowed, sequence evidence given up. Raptor X is the worked instrument, and its p-value and GDT score are how the question, "Does this fold accept me?" becomes a number. This section explains what ab initio structure prediction claims to be, and what the chapter actually puts on the page. Ab initio means the energy function is the only oracle. You fold the chain by minimizing physical forces over conformations, consulting no previously solved structure. That principle is stated flatly in section 31.1, and the chapter's own worked example undercuts it. Raptor X partitions. Bubeline dicer. 1 into 6 domains depending on the available template structures in the protein databank. It then grades the result by alignment score, by useek-ed, and by a p-value against random. Every one of those quality metrics is inherited from template alignment. The slot this chapter occupies in the book's arc, with threading in chapter 30 and validation in chapter 32, is the no homologue fallback rung. The tool placed on that rung remains homology-driven. Read the chapter as a Raptor X protocol whose title names an ambition the procedure never exercises. This section explains why a predicted protein structure has to be validated against empirical geometry rather than against an experiment. A predicted structure arrives as coordinates with no evidence attached, and validation supplies the missing likelihood. No crystal exists to compare against, so the model is scored against what real backbones actually do. The Ramachandran plot compresses the phi and psi angles from 121,870 residues across 463 solved X-ray structures into a decision surface. A residue landing in the white region occupies a sterically forbidden rotation, and that convicts the model rather than the plot. Glycine sits separately because, lacking a side chain, it escapes the very constraint that makes the surface informative. The errors and warnings issued by WHHF run the same logic over the remaining geometry. Validation is an empirical prior standing in for the experiment. The prediction was built to avoid. This section explains how transcription factor binding sites are predicted. A binding site is a distribution over strings. Transfac stores that distribution as a position weight matrix, 1 weight per nucleotide per position, and MatTrack slides it along a sequence, scoring core similarity and full matrix similarity separately. Prediction reduces to convolving a probabilistic pattern against a string. Everything else in the chapter is interface. The price of that tractability is stated in the opening lines and never revisited. Each position contributes independently, and motif length is fixed. Cooperative base contacts and variable spacer motifs, therefore, sit outside the model by construction. Its place in the book's arc is the turn where sequence annotation stops asking what is here and starts asking how well does this match a curated prior. This section explains how translation initiation site prediction is set up as a problem and why every method for it has the same shape. Every ATG in a transcript is trivially findable. Which one the ribosome actually uses is not. Kozak's scanning rule makes the first AUG the default answer, and leaky scanning, reinitiation, and internal initiation are the exceptions that turn a lookup into a classification. That is where the chapter's shape comes from. Score the fixed window of flanking bases around each candidate ATG, apply a threshold, then rank. NetStart runs a neural network over that window, and TIS Miner runs a support vector machine. Kozak's weight matrix and Salzburg's conditional probability matrix are the same object at lower capacity matrix are the same object at lower capacity, only the scorer changes. The feature is always the neighborhood and never the codon itself, which is why the Kozak consensus survives as a column in TIS Miner's output rather than as the decision. This section explains what molecular docking computes and what it hands back. Binding becomes a minimization. Docking asks whether two molecules interact by searching the space of relative placements and scoring each by free energy. The pose at lowest delta G is the prediction, and the lock and key picture is just that landscape having a sharp enough minimum. Everything downstream follows from the split. This induces a search that proposes poses and a scoring function that ranks them. That is why the chapter's procedure is mostly preparation. Strip waters, pick one cavity, hand Swiss stock, a clean receptor, and a MOL2 ligand. It's 1E, IO. Run returns 5 poses from minus 10.77 to minus 8.75 kilocalories per mole, with the top two apart by 0.01. So the deliverable is a shortlist someone else must adjudicate. This section explains what genome annotation actually delivers when the genome is prokaryotic. Annotation here is assertion of coordinates. Given a naked prokaryotic contig, Yersinia pestis CO92 in the chapter's worked case, genome becomes a set of labeled intervals, a table whose entire content is gene index, strand, left end, right end, length, class. Everything downstream, the translated protein and the extracted nucleotide sequence, is a projection of those integers back onto the string. What makes this cheat in prokaryotes is the absence of interruption. An open reading frame runs uncut from ATG to a stop, so the boundary problem collapses to finding where a Markov model's coding statistics switch on and off. That is the chapter's real position in the book's arc, the easy half, deliberately shown first, so the eukaryotic case that follows has something to break. This section explains why eukaryotic gene finding is a parsing problem rather than a search problem. A prokaryotic gene can be found by scanning for an open reading frame. A eukaryotic gene cannot because it is split. So Chapter 37 replaces search with parsing. GenScan treats the sequence as a string emitted by a hidden Markov model whose states are the gene's grammar, promoter, initial exon, intron, internal exon, terminal exon, poly-A signal. Annotation becomes recovery of the most probable state path. Every column of the output table is then a score under that grammar, splice site strength, coding region likelihood, exon probability summed over all passes. The chapter's cautions fall out of the same structure. Mask repeats first, and trust internal exons over terminal ones, since an exon bounded by 2 splice sites carries twice the constraint. This section explains how real-time PCR turns concentration into a clock reading. Doubling makes template exponential in cycle number, So the cycle at which fluorescence crosses a fixed threshold, written CQ, carries the log base 2 of the starting quantity. A 10-fold dilution shifts that crossing by exactly 3.32 cycles. This is why the slope of the standard curve measures amplification efficiency rather than anything about the slope of the standard curve measures amplification efficiency rather than anything about the samplet. The chapter's payoff is that a logarithmic readout turns division into subtraction. Livak's method, 2 to the power of minus delta delta CT, first subtracts a reference gene to cancel loading, then subtracts a calibrator sample to cancel gene. Specific efficiency. Fold change falls out of 4 numbers with nothing calibrated in absolute units. One saying, the plateau phase, where reagents exhaust, carries no quantitative information and is discarded. This section walks through why microarray analysis needs the apparatus it does. Hybridize a labeled sample to 40,000 immobilized probes and read the light. The measurement itself is a photograph. The chapter's whole apparatus exists because that parallelism is also the problem. Cowdery's sidebar carries the compression. A replicate correlation of 0.97 means 3% random variation, and 3% of 40,000 genes is 1,200 false positives before any biology enters. Run it in duplicate, and 0.03 squared leaves 36. Every stage that follows is one move, applied repeatedly. Background subtraction, normalization against psi3 and psi5. Incorporation bias, Bonferroni's 0.05 divided by 20,000, and the retreat to FDR once that threshold admits nothing. Reading many channels at once, that threshold admits nothing. Reading many channels at once shifts the science from detection to error accounting. Chapter 39 is the book's argument that scale changes what statistics must do. This section explains what single nucleotide polymorphism mining tools actually do. SNEAT mining is a filtering problem disguised as a discovery problem. The polymorphism is already sitting in the sequence pile. What the tools do is separate real variation from sequencing error, and both look identical in a single read. The chapter's move is to make that separation statistical rather than observational. Redundancy is the discriminant. A base that differs from the reference in many independently sampled reads is a variant, and one that differs in few is noise. Every tool in the catalog is a different threshold policy over the same alignment pile. The consequence generalizes past SNPs. Any in silico discovery over noisy reads is a signal-to-noise decision, so the tool's real specification is its false positive posture. A feature list tells you nothing about where that threshold sits. This section explains why a microsatellite counts as a mutation clock, and why finding one is a text problem before it is a laboratory problem. A microsatellite is a mutation clock that leaves its reading printed in the sequence itself. Slip-strand mispairing adds or drops whole repeat units far faster than point mutation, so the count n in the repeat AC taken n times varies between individuals while the flanking sequence stays fixed. A locus becomes a marker the moment you know where the repeat starts and where one how is in day. The chapter's whole move is that this is a text problem. The program misa.pl reads a FASTA file and reports one mono, repeat 27 times over at positions 802 to 828. No gel, no primer, no organism. Mining precedes wet work. Primer 3 only afterwards converts the coordinates into something amplifiable. The marker exists as an interval before it exists as a reagent. This section explains why every RNA sequencing pipeline has the same shape and what that shape costs. Reads are cheap, and reference genomes are large, so every pipeline does one thing: turn a pile of short strings into per gene counts, then make those counts comparable. Both halves are approximation forced by cost. Exact pairwise alignment by dynamic programming is unaffordable at next-generation sequencing scale, so aligners index the genome instead using Burroughs-Wheeler plus the FM index or hash seeds, and they pay for that speed in sensitivity. Raw counts are equally uninterpretable, since a longer gene and a deeper library both inflate them for reasons. The chapter's whole content is those two divisions: divide out length, divide out depth. Everything downstream, differential expression and annotation alike, inherits whatever those two approximations distorted. This section explains what the chapter does with 4 differential expression callers that disagree. The first move treats that disagreement as signal and keeps only the intersection, 4,246 genes out of roughly 10,800 called by at least one caller. The second move converts names into meaning. A gene list says nothing by itself, so each gene ontology term is scored by overrepresentation against a species background. 210 of your 4,133 genes, hitting a 633 gen intertron 1000 la mounted, hooping converts one caller. Both moves are the same normalization. A raw count stays uninterpretable until divided by its expectation. Which tool performs it? G. Profiler, David, or Clue GO is interchangeable plumbing. The chapter stops where the method does. Interpretation of the data is completely the researcher's purview. This section explains what the differential expression chapter is actually doing. Nothing in this chapter tests a gene. Every test compares 2 conditions through a model of what counts as noise. The whole apparatus, Princeak light filtering, G-snap mapping, SAM tool sorting, exists to deliver a 2-condition table to one statistical decision. The 2 roots taught split precisely on what fills that table. Cufflinks, hands cuffdiff, FPKM abundance already normalized by library size and gene length. RCM hands EBSeq, DESeq2, and EdgeR raw counts normalized inside the model. Same reads, 2 gene lists, because the normalization choice is itself the inference. Replicates are the chapter's one non-negotiable. Cuffdiff, run on a single sample per condition, has no variance to test a fold change against. This section explains how MeRDeep2 finds and measures microRNAs. A microRNA is defined by how it is manufactured, so detection scores reads against biogenesis rather than against a sequence library. MeRDeep2 folds each locus that reads pile onto, then asks how closely their distribution over that hairpin resembles what Drosha and Deser would have left behind: mature arm dense, star arm sparse, loop empty. A probabilistic model turns that resemblance into a score carrying its own statistical controls. The chapter splits the work 3 ways. Mapper.pl collapses and maps the reads. Quantifier.pl counts reads against known precursors. MeRDeep2.pl scores candidate folds for novelty. Expression is simply the count of reads that survived recognition. The structural test and the measurement are one pass. This part explains why microRNA target prediction cannot be done by base pairing alone. A 7-nucleotide seed match occurs by chance every few hundred bases, so complementarity alone cannot locate a target. The entire chapter is what gets stacked on top of that insufficiency to recover specificity. Each tool is one weighting of the same shortfall. Target scan buys confidence with conservation across mammals. TixRNA target buys it with site accessibility, the unpaired energy needed to open mRNA secondary structure. Miranda buys it with a weighted dynamic programming score that tolerates wobble pairs between G and U. The chapter's second move is that the relation is symmetric and indexed both ways. The HMG, a 2 query, asks which microRNAs hit one gene. The MyR13P query asks which genes one microRNA hits. Same table, read from either side.